As you all know, bryophytes are an important group of plants, also known as amphibians of plant kingdom. This group is having the distinction of being the first land plants. When we look on the life cycle of bryophytes, we come across two distinct generations, gametophyte and sporophyte, with gametophyte being dominant in the life cycle. The gametophyte in bryophytes is haploid, green and independent, while as sporophyte is diploid and dependent physically as well as physiologically on the gametophyte. In the life cycle, gametophyte bears gametes which fuse to form the zygote, which develops into sporophyte, which after development undergoes meiosis to form spores which give rise to the gametophyte again. As far as origin of sporophyte is concerned, there are two conflicting theories concerning the origin of sporophyte in bryophytes. One is called as homologous theory and the other is antithetic theory. First, I will explain homologous theory. The homologous theory is also called as transformation or modification theory. This theory was initially proposed by Pringsheim and later it was elaborated by Frist. According to this theory, the sporophyte in bryophytes originated by a direct modification of the gametophyte and took the specific function of spore production. The sporophyte thus originated was similar and homologous to the gametophyte, and both were free living and photosynthetic. The evidence in favor of this theory is the occurrence of similar but distinct photosynthetic sporophyte and gametophytic generations in some algae such as Ulva, Cladophora and Ectocarpus, which are believed to be the ancestors of bryophytes. Thus, in the first land plants, the gametophytic and sporophytic generations were independent and isomorphic. I mean, they were similar, but gradually the sporophyte became dependent, less complex and attached to the gametophyte. The other strong evidence of homologous theory or direct modification is the phenomenon of apogamy and apospory occurring in present-day bryophytes and pteridophytes, where sporophyte develops directly from gametophyte without involving gamete formation and fertilization. The homologous theory thus believes that sporophyte in bryophytes originated as a result of direct modification in the gametophyte. The second theory is antithetic theory. This theory was first proposed by Salvosky in 1874 and later it was strongly supported by Strasburger, Bauer, Chamberlain, Campbell and Cavers. According to this theory, the gametophytic generation is the original one and sporophyte was added to the life cycle intercalated between two successive gametophytic generations. This theory suggested that the sporophyte originated from the zygote by undergoing mitosis and elaborate development. The algal ancestors of bryophytes produce gametes which fuse to form the zygote, which soon or after a period of rest divides meiotically to produce haploid cells which give again rise to the gametophytes. Thus, the sporophyte in them is simply represented by the unicellular zygote. However, in bryophytes, the first sporophyte appeared when the gametophyte evolved orchigonium and then retained the zygote. The retained zygote underwent mitotic divisions instead of meiosis. This resulted in a mass of diploid cells forming a multicellular structure, the ancestral sporophyte. Successively, the cells of this cellular body divided meiotically to produce the haploid spores. This represented the simplest multicellular sporophyte of bryophytes, in which all the cells were fertile, besides being very much different from the gametophyte morphologically. Later, the progressive sterilization of this ancestral fully fertile simple sporophyte led to the evolution of complex sporophyte in bryophytes. A sporophyte with maximum fertile tissue and least amount of sterile tissue is the simplest and most primitive, while as the sporophyte with maximum sterilized tissue and least fertile tissue is highly advanced. This maximum sterilization also develops complex structures in the sporophyte. The complex and more advanced sporophytes have evolved from such simplest primitive sporophyte by progressive sterilization of the fertile tissue. According to this theory, the most primitive and simplest sporophyte is found in the genus Rixia, which has evolved from an ancestral sporophyte that consisted of a mass of sporogenous tissue with all cells being fertile. The first step in the evolution of sporophyte involved the sterilization of outer layer of sporogenous tissue, which resulted in the formation of a jacket layer around the sporogenous tissue. Such a sporophyte consisted of a spherical capsule devoid of foot and seta. This type of sporophyte is present in Rixia glauca. The evolution of sporophyte is seen a step ahead in Rixia crystallina, where all sporogenous cells are not fertile and spore producing. Some sporogenous cells fail to form spores and thus are sterile. They are called as nurse cells and serve as nutritive cells. The stage in the evolution of bryophytic sporophyte involved the formation of foot and seta. These structures came at the expense of the enhanced sterilization of the potential sporogenous tissue of sporophyte. The occurrence of foot is first seen in Corysnea, where basal part of the sporophyte is sterilized to form the foot. The sporophyte thus consists of a small foot and a capsule. The capsule contains a single layered wall which encloses the sporogenous cells which produce the spores. There is also sterilization inside the capsule with many potential sporogenous cells forming nurse cells. A step further in evolution is the sporophyte of Spherocarpos, which is organized into a small bulbous foot, a short seta and a capsule. Spherocarpos is thus having additional sterilized tissue in the form of seta, which is 2 cell thick. Targonia sporophyte displays a further step of progressive sterilization of potentially sporogenous tissue or fertile tissue. The sterile regions of the sporophyte comprise of a broad foot, a well-developed seta and a large number of elaters in the capsule. Half of the potentially sporogenous tissue in the capsule is transformed into sterile elaters with 2-3 spiral thickenings. The next stage in the progressive sterilization of sporogenous tissue and evolution of sporophyte is seen in the sporophyte of Markantia. Here the hypobasal region of the embryo forms the foot and lower part of seta, while as the epibasal part forms the upper part of the seta and the capsule. The capsule is having a single layered wall derived from epithecium, which becomes multi-layered at the apex of the capsule. This multi-layered apex splits halfway down into many segments at maturity, thus helping in dehiscence. The endothecium gives rise to archosporium, which forms the sporogenous tissue. Only half of this sporogenous tissue produce spores, while as the rest remain sterile and transform into elongate elaters. Some sporogenous cells at the top differentiate into sterile apical cap. Thus, the sporophyte of Markensia is having a foot, a seta, elaters, several layered apex and apical cap as the sterile tissue. Here the additional sterile tissue is exploited for capsule dehiscence and spore dispersal. The next higher stage in sporophyte evolution and structure complexity is met in the members of order Zingermanials. In Pelia and Riccardia, the sporophyte differentiated into foot, seta and capsule develops from the epibasal half of the zygote only. The hypobasal half of the zygote does not play any role in sporophyte development. The wall of the capsule is 2 to 8 layer thick with thickening bands and is derived from amphithecium. Here also the entire endothecium produces archosporium, which by repeated divisions gives rise to sporogenous tissue and elaters. Further sterilization of sporogenous tissue gives rise to the formation of an elateriophore, which occurs at base in Pelia and apex in Riccardia. Thus, further sterilization or reduction of sporogenous tissue results in new structure with more efficient mechanism of spore dispersal. Further sterilization of sporogenous tissue is found in the highly specialized sporophyte of Anthoceras, where more specialized structures develop at the cost of reduction in sporogenous tissue. The sporophyte of Anthoceras consists of a long cylindrical capsule and a foot with a meristematic zone at its base. The capsule wall in Anthoceras is multilayered with stomata and air chambers. The cells contain chloroplast and thus the capsule wall is photosynthetic. The capsule wall develops from the outer layer of epithecium, while as the inner layer of epithecium gives rise to spore sac. The sporogenous sac contains spores and pseudoelaters. The whole endothecium is transformed into a column of sterile tissue of elongated cells called as columella. The sporogenous tissue is restricted to a small portion of the capsule which overarches and surrounds the central columella. The ultimate stage of sporophyte evolution in Bryophytes is seen in the mosses such as Polytrichum and Funaria, where progressive sterilization of sporogenous tissue has reached its maximum. In these sporophytes, the sterile tissue is specialized into varied and complex structures which perform diverse functions. Thus, the highest degree of structural complexity in these sporophytes is directly the outcome of maximum amount of sterilization of sporogenous tissue. The sporophyte of Funaria is differentiated into a sterile bulbous foot, sterile long seta and a complex capsule. The sterile capsule wall, which is multilayered and photosynthetic, is derived from amphithecium. The endothecium gives rise to archosporium from its outer layer only, while the rest of it develops into a massive central sterile columella. The extremely reduced archosporium consists of a single layer of barrel-shaped cells and is held between operculum and apophysis. Thus, in Funaria, major portion of sporophyte is sterile, and the sterile structures include foot, seta, capsule wall, apophysis, peristome, operculum and columella. This structural complexity has evolved for an efficient spore dispersal mechanism. The fertile zone in this type of sporophyte is restricted to a much smaller portion. Thus, this theory conceives moss sporophyte with maximum complexity and sterilization as highly advanced and believes its evolution from Riccia by progressive sterilization of fertile tissue. This theory is also called as theory of progressive simplification or retrogression theory. This theory is supported by Kashyap, Church, Goebel and Evans, who believe that the evolution of sporophyte in Bryophyta has occurred through reduction or progressive elimination of sterile tissue. According to this theory, the sporophyte in Bryophytes having maximum amount of complexity and maximum amount of sterilization is primitive, while as the sporophyte with least amount of sterilization and least complexity is the advanced one. Applying this principle to Bryophytes, we see huge amount of complexity and huge amount of sterilization in the moss sporophyte such as Funaria and Polytrichum. Thus, according to this theory, the moss sporophyte is conceived to be primitive, while as the Rixia sporophyte having least amount of sterilization and complexity is believed to be highly evolved or highly advanced. This theory holds that the advanced sporophyte of Rixia has evolved from complex and elaborate sporophytes of mosses such as Funaria by progressive elimination of sterile tissue, retrogression or progressive simplification. According to this theory, the sporophyte of Rixia has evolved from the sporophyte of mosses along the following lines. First, the sporophyte of Bryopsida such as Funaria and Polytrichum are most primitive because of their highest amount of sterile tissue and complexity. Then the second step involved the peristome and peristome facilitated spore dispersal has been eliminated as we find it in sphagnum. I mean, there is gradual simplification and elimination of dispersal and dehiscence mechanisms. The third step which led to the evolution of advanced sporophyte of Rixia is the photosynthetic tissue in the capsule wall has reduced gradually and ultimately eliminated from the wall of liververts such as in Markensia and Rixia. The fourth stage involved the multi-layered wall gradually changed into a single-layered wall together with elimination of stomata and air spaces in wall layers, for example, as we find in Rixia and Markensia. The fifth stage led to the gradual disappearance of foot and seta, resulted in a sporophyte which remained completely embedded in the gametophyte thallus, for example, Rixia. This type of reduced sporophyte with little sterile tissue and maximum fertile tissue is regarded as most advanced according to this theory of sporophyte evolution.